Yep, just take a seat wherever you like. It's hot in here. Gonna wait a few more minutes, let people kind of trickle in. It's first day. People are, I'm sure you know, still trying to find their way to the classroom. So yeah, it's kind of down in it. It's like in the area that talks about the fact that we have lectures. That's where it has the room number. So they have dictated now. They've been actually pretty strict about it.
Yeah, they've been strict. They have to have like a specific format with the syllabi now.
Yeah, it's very strict now. So there's not a spot for putting in the classroom, so I just shoved it in there. So yeah. Yeah.
Wow.
I mean, I didn't change this the format, so
Yeah.
but yeah, they changed the syllabi.
I used to hang out in the dean's office last year because I a lot to people, and I think they were talking about the format thing.
So I usually go by the time it's on like iPhones and stuff. I figure like that's more official than the crap that you'll see on these. Whatever's going on with these most of the time, they're not accurate. So if you are running late, then you know if you're looking at your iPhone, it says you have 3 minutes. Even if this doesn't say that, then you got 3 minutes. It's not like I count people off for being late, but if you come in late into lecture, just be sure you've signed the sign-in sheet. I'm going to pass around the sign-in sheet here in a minute. But the sign-in sheet is what I'm going off of for your attendance points. There's points tied with attendance. So if anybody in here has actually looked at the syllabus and read over it, and you've probably also seen that it's not just attendance points, it's participation points too. So we will be calling on people by name in this class and asking you questions. It's not going to come out of nowhere. Luckily, we have in our lecture, I'll have it set up about halfway. And then again, at the end, we'll have calling on people for questions and you give me the answer. There will be kind of, I want to say, a reward system based on that as well. Some days I'll be calling on you by your name and you'll be expected to answer the question. Other days, I will be calling on your group. Okay. So today, obviously, it's going to be by name because some people may not know that they have a group today. You do have a group. If you haven't been on Canvas, you really should be on Canvas by now. You will see what group assignment you're assigned to. Everybody should have a group by now. This class is full. We have 24 out of 24 students in this section. So that's how I know not everybody's here because I only have 24 chairs set up and not all of them are full. So but yeah, there is credit associated with this. If you are somebody that shows up to class every day, but you refuse to answer the questions when I call on you, I'm going to remove your credit. Okay, because it's participation based too, not just for showing up. If you show up and you refuse to answer the questions, then I'm going to be like, why are you even here? Like literally why? The questions aren't here to and when we get to them, I'll remind you guys, but they're not really here to make sure you're getting them right. Okay, they're here to get you guys thinking about the material. So if I ask you a question like, what is the material that makes up bacterial cell walls, which we're not getting to today, okay, and I call on you, and you're like, you don't know, well, I would suggest trying to make a guess. Like, is it like sugar-based? If you vaguely remember there's something to do with sugars or like proteins or something, like you vaguely remember that, then yeah, I'm gonna probably give your group a point. So these are all group-based points. So when I call on you as an individual, if you are in group alpha, and you get your question right, group alpha gets a point. So that's why there's even more stakes on this to pay attention during the lecture, because it's all going to be what we just talked about in the lecture. All of the questions will be, okay? So halfway through, it'll be questions about what we just in the first half talked about, right? It's only 5 questions in each time than I call people. So it's not like we're gonna go through them for like 20 questions and wish you were dead and all of this. But it's just to make sure we're paying attention. Because, I mean, if you guys even try to tell me that you're gonna be able to sit through every single lecture in this class without zoning out, I will call you all liars, because we all know how this goes, right? It's 10 a.m. on a Monday morning, like, who's gonna actually sit in this class and listen to me blab on and on for the whole class period without zoning out? That's what the point of the questions are, just to make sure you guys are actually like paying attention and then engaging you with the material as soon as we are hearing about it, so that it will help with retention. If you get the question wrong, I'm not going to be like, wrong, like you're terrible, you're dumb, you don't know anything. That's not how this goes. I actually feel like sometimes whenever people are making an educated guess, like if they're like, I ask the question about the cell wall and you're like, is it like plant cell wall, cellulose? Well, that's an educated guess. I mean, you're basing it off something you know in science. It's wrong, but it would be an educated guess. So I'm not gonna be like, well, you're totally wrong, sit down. Anybody else really know the real answer? Like, that's not how this is gonna go. What I will do is, first of all, if you get it wrong, I'll open it up first to the rest of your group. So if you're in group alpha, I'll open it up to group alpha. Anybody in group alpha can then answer that question and see if they get it right, right, okay? If somebody in that group then can make an appropriate guess based on the information we've been talking about, then you'll still get the point, even if it's not completely right, just for having been paying attention. So that's what this is more about. It's showing that you've been listening. And then you'll get points for that. Those points from your groups, they all go towards your extra credit. So we only have 3% extra credit. That's how every science class has been for years now. You can only have 3% extra credit. And 1% of your extra credit will be this crap, me asking you questions, okay? There's another 1% that is going to be associated with having completed everything in the course. Either you complete it or you don't. And then you get your 1% or you won't get any of it. There's no gray area in this one, okay? If you miss an assignment and you had a reason for missing it, a verifiable reason, and you guys know what I mean, a doctor's note or something like that, where somebody can contact me verifying that, hey, I had to keep her at work for whatever reason, you know how it goes sometimes, then yeah, I'll let you make it up. But if you're just missing it, or like, I have no reason to think that the excuse that you're giving me is verifiable, I can't give favoritism, right? Can't just say, oh, well, I like you, and I'm sure you were doing this thing, but what about the other person that also didn't have a verifiable excuse? Do I give everybody an excuse now from now on? And so that's why we have that rule, okay? Anyways, so whatever. For the attendance, for whatever assignment it is that you miss, if you have a verifiable reason for having missed it, I'll typically let you make it up, okay? And then that can be fine and count towards you having completed everything, and you can get that goal of getting that extra credit, okay? Yeah. But yeah, so that's kind of where I was going with the question thing. That'll be part of 1% extra credit is that, 1% of the extra credit is completing everything in the course, and then the other 1% is going to probably mostly tied to, we already know it's syllabus quiz and the integrity statement. If you've seen Canvas and you had to open up your modules, which is where you will start. You start there. If you've already done that, then you know what I'm talking about. If you have completed your syllabus quiz and your integrity statement, then you probably have 0% in the next class. And if you've completed nothing else, you will. That's okay. That's not real. I hope you guys understand that's not real. Okay. Once you have at least one assignment in there, it'll populate with that as your grade. And then it will, of course, like normal grades do, go from there. So don't freak out if you do that extra credit something. It's just like trying to add the extra credit to something, but there's nothing. So it makes it 0, and it's not real. So don't freak out. Come on in, you're fine. You can sit with. But yeah, if you come in late after we get started, just be sure you have signed in on the sign-in sheet, because that's going to be your attendance. All right. Most of the stuff, as far as like what assignments we have, how many points there were, what's expected of you as far as lecture versus lab, I expect you guys to go on Canvas and actually read your syllabus and actually go through the video that's associated with the course to make sure you can understand what's going on in the course. So I'm not going to go over it here. It's just, we can read, right? So that's what I'm going to touch on. But yeah, so I'm going to pass the attendance sheet around. Be sure you sign it or whatever. Put your mark, if you will, and be sure that if you are here every day that you have signed that sheet before you go. If you come in late and you forget about it, come to me at the end of class and be like, I forgot to sign the sign-in sheet. I'll let you sign it. you were here, I'll count it, okay? So I'm not that big on like, you have to be here at a specific time. I don't like coming in this back door back. Because I can't read on. All right. I find there's another door over here. Okay. Yeah, sorry, I can't unlock this. Anyways. So we'll pass around that sign-in sheet. Make sure you sign in every day. That'll be your attendance point. As far as what we do here in class, typically every day I will start with a Kahoot quiz. Not today, because we didn't have a class before this one. So I'm not going to do it, but typically every day we'll start with a Kahoot quiz. So typically every day you should have laptop or phone or whatever it is that you would use to get online to take a Kahoot quiz. It's on an actual website. I do have a QR code that'll pop up, or it's like an actual little number code, and you'll just go to kahoot.it and then enter in the code, and it'll join you in with everybody in here. We will play it live all together. Okay. These Kahoot quizzes are also available on Canvas. You can play them on your own time. If you want to play them multiple times, like if you want to play them like in the beginning of having learned the chapter, and then later on when you're studying the whole unit, that's fine. Just use a different username, maybe a different nickname or whatever. So you can use it. You can do it as many times as you want to, as long as it's open. So. But we'll usually start out with a Kahoot quiz. Like if we are in the next one, which will be chapter 2, right? Then chapter 2 will have a Kahoot quiz over chapters 1 and 3 to review that before we get started. Okay. So we'll review the previous chapter with a Kahoot quiz. It's only 10 questions. We'll run through that, and then we'll start in with the lecture. And remember, halfway through the lecture, 5 questions where I'm going to call on you. At the end of the lecture, another 5 questions where I'm going to call on you. Today, I'm literally calling on you by name. And then other days I'll just pick your group, not always, like we'll just switch it up and see which is which. And then anybody from the group can answer. If we start having issues where like I call on group alpha and nobody answers, then I'm going to go back to calling on names. Okay, so just be aware, like sometimes it might be better just to talk up and not have to go through that. The groups are assigned on Canvas. You can find what group you're in there. It's either Alpha, Bravo, Charlie, Delta, Echo, or Foxtrot. Those are all phonetics for A, B, C, D, E, F. Right, so it's like if you, anybody in here ever work in a hospital, you have to actually call other people and talk about patients or anything like that. If you have to verify a patient name, the proper protocol is to use a phonetic alphabet when you're having to spell out something. That's a proper protocol. Not everybody does it. I used to work at the HLA lab, which is the transplant lab at Integris. And so if we had like an organ coming in for a patient and they say a name like Smith, and like we want to be sure we're getting the first name right. So we might actually spell it out phonetically over the phone because it's easier to tell the difference between Bravo and Delta than it is for B and D, right? So that's where the phonetic alphabet comes from. You guys haven't known. It's not a military thing necessarily. It's they use it a lot, but it's not just military. So
Sorry, man,
be aware of that. You're okay. So do you know what class you're trying to go to?
Human anatomy?
Upstairs.
Upstairs?
It's on the 3rd floor, like right at the end of the hallway.
Yeah, all the way on the other end.
Okay.
Leave the elevator, I think, go to the left.
Yes. So, so you'll have groups A, B, C, D, E, F, basically, and they're just named after the phonetics. Those groups, yes, they apply whenever we're asking the questions here in class. It's just so we're not left alone answering questions, right? So we don't get left alone that way. The other thing is for your projects, for the group projects that we have them for every single unit, and each group is assigned a topic from that unit. So Alpha, I think, is assigned like germ theory, which we're going to talk about today, and then so on and so forth for Unit 1. And then you guys are meant to basically, they're like a review project. So you can do it as a poster or a presentation, a handout or a quiz, or you can act it out as a skit or whatever it is, but whatever your topic is, it will act as a review for the rest of the class over your topic, right? So the idea is when you do that, you and your group should be pretty well versed in that topic at least. So at least you got that one down, and hopefully it'll help other people learn through a different method of learning that topic. So that's the hope with those. I also have shorts that I have, audio shorts that I've recorded. I don't know if I sent out a notice about that, but the audio shorts are like 10 minutes or less. Just me talking about whatever that topic is for that short. So there's like one about, bacteria. And so you would like listen to me talking about like what bacteria are and their characteristics for 10 minutes. Instead of like having to listen to an entire lecture over a chapter, I'll cover the gist of a major topic for 10 minutes. It's good if you are a visual learner, seeing all the slides and everything like this when we're in lecture, But when you're reviewing, maybe you just want to review one topic, go listen to the audio blurb. Or if you're not this overwhelmed, having visual and audio and all these questions and everything like that, that might help you pick up the topic a little bit better than just relying on, lecture. So it's there for your use if you guys want it. It's in a big file at the end of the Unit 1 module. Any questions about that stuff? Yeah. Other than that, like you have a group, yeah, but study with whoever you want to outside of that. Like you're not like required to only work with your group ever. Like that's not it. And you don't have to stay with your group in lab. I don't care who you work with in lab. Okay. So when we do get to lab next week, you can sit with whoever you want. I don't care. Okay. But in here, you'll have your that group. There is no switching with anybody in groups. You are assigned a group. The idea is, I know for some of you, you work at jobs and you already know that you have to get along with people that you don't like. Some of you may not have experienced that before, but this is an idea of you will sometimes be thrust into a job. You think you're going to go work at this hospital and you are working with all these people that you don't know, and you have to learn how to get along with people, right? And work with them, even if you don't like them, even if you don't want to. That's being an adult, right? Having a job, right? So that's the idea with those projects. There's only 3 projects. So hopefully I'm not dragging you too much through the mud there, because I'm not meaning to. It's meant to be beneficial, right? If you have somebody in your group, and I doubt this will happen because I've never had any students like this before, but if you happen to have somebody in your group who is just adamant, like definitely not going to participate in your group, they're just like, yeah, I'm not doing this crap. Yes, you have a question. Okay, right at the bottom for me. Yeah, because some people have switched in and out. So I printed that out like 4 days ago. So I don't know, it's been a little while, but anyways, yeah, just right at the bottom, and I'll get it added in for the next one. I can't remember what I was saying. I get so scatterbrained. Thank you. Yeah, if you have somebody in your group that just really is not participating, or like they'll just sit there and like not talk with you and like expect to get the grade for the thing, you need to let me know, number one, if that happens, if it's that bad, right? And if you have somebody that really is like they're showing up but they just expect to like do the absolute bare minimum, we will be grading each other on those group projects as well. At every single group project, you'll be turning in an anonymous rating for everybody in your group, including yourself. That way I don't know who's rating who. But yeah, we'll get to that when we need to. Okay. So don't worry. Have fail safes there.
Is the syllabus really time group? No, we're in.
It is on the announcements page.
Okay.
On oh, I tied my hair into this shirt. On the announcement page on Canvas. Also on Canvas, if you guys aren't getting notifications when I set up announcements or something like that, be sure that you do. I send a lot, first of all. Second of all, if there's a change or I'm adding on something, or I'm letting you know, like if there was like a weather day or something like that, or somebody's had like a question that several students have had, and I want to be sure everybody knows it. Typically, I'm going to post it in the announcements. So you do want to be sure that you're keeping up with those. So FYI, announcements are pretty serious. All right, let's do this. Yes, what's the question? Oh, yeah. If you want to just come set it up here, that way if anybody comes in late, they can. Thank you. Yeah, if you guys are the last one that gets the sign-in sheet, you can, yeah, you can always come set it up here at the front. So let's go ahead and get started with our lecture today. I don't know if you guys noticed, but I started up Zoom. I Zoom every single lecture. If you are somebody that can't be here because you're sick, or hey, maybe you're just somebody that learns better sitting at home. I don't know. It counts as being here if you're on Zoom. Okay, that is attendance, but I can still call on you if you're on Zoom. Okay, so I expect you to have like a microphone and everything ready if you're on Zoom to be able to participate with the rest of the class. But it's fine if you want to do Zoom. All right, I have it live and I also have it recorded, and I usually will post the recording every time that we're done. So you have that to look at afterwards too. All right, so we'll try to run through chapters 1 and 3. It's a lot, so we're going to try to push through a little bit. They're basic stuff, but I don't want to miss anything important. So microbiology is basically a specialized area of biology, looking at organisms that are micro, too small to see with the naked eye. So we need, magnifying glass at the very least, up to proper microscopes, maybe even electron microscopes, in order to see these things. So we have cellular and non-cellular organisms when we're talking about these microbes. The cellular ones are going to be our bacteria, archaea. Bacteria and archaea are very similar in what they look like and how they're structured and how they kind of divide and everything like that, but their chemical makeup is very, very different. So bacteria and archaea, then we have fungi, we have protozoa, we have helminths, which we would consider to be microbes, even though we know, we probably know this, that when we talk about infectious worms, like parasitic worms, like heartworms, even like we have in dogs, or pinworms, like little kids can get, you know, I don't know if you guys know about pinworms, but little kids can get little itchy booties and they scratch their butts, and sometimes you put tape over it to see if there's worms there. If you haven't heard of that, that's an actual thing here in the United States. It's pretty common. You can even buy medication to treat it over the counter. So those are helminths. These are going to be the parasitic worms. A lot of these, you don't need a microscope to see them, but their eggs and their larvae, which is what we often will use to diagnose them, because the bigger worms often stay inside of the body and we can't see them as easily. That will be what we can use to identify them. So those are micro. Right? All right, so the non-cellular organisms, these guys do not have any cells. They're not made of cells. They don't have the structure of a cell. They're not cells, okay? And that's viruses and prions, okay? Viruses, that's why we have arguments all the time about whether viruses are alive or not. If you guys don't know that, that's a thing. They're not cells. They cannot live outside of a host cell. They need the host cell in order to, you know, reproduce and do anything at all. They can do nothing without the host cell. So they don't have all the parts that cells normally have. So they just are kind of on their own. Viruses are weird. But yeah, so remember that. Non-cellular, viruses and prions. Just think that, and the other ones are all cells, okay? All righty, so we have eukaryotes versus bacteria and archaea. So eukarya, eu means true, karyote means nucleus, okay? So true nucleus, that's going to be anything that has a nucleus, and anything else that is membrane bound. We're going to get into this in chapter 5 in more detail, but membrane bound, like mitochondria. You might have heard of those guys, right? Mitochondria, endoplasmic reticulum. They're all organelles. They're membrane bound structures in a cell. Only eukaryotes have them. So that means if I'm saying versus bacteria and archaea do not have them. They don't have membrane bound anything. They have just their cell is membrane bound and everything else is just free floating in there. Okay? So bacteria and archaea do it that way. They are also known as akaryotes. So A being like without, and karyo being nucleus, so they don't have a nucleus. Okay. If I were to have described to you just now, which I did, bacteria and archaea as having no nucleus, and they're just really like just a whole bunch of crap just floating around inside of a cell, right? Contained, doing whatever it wants. And then I told you that the eukaryotes, like us, our cells, have little compartmentalized structures that have, you know, set uses and set roles in the cell, right? Which one of those sounds more evolved, more, you know, complicated? That would be the eukaryotes, right? They've got more structure going on, everything like that. That makes sense when you think about it that way. Well, we think that, of course, the akaryotes, like bacteria and stuff, came first. And we think that what happened was some other cell of some sort of stage of evolution kind of ate up a bacteria, but they found out that they were symbiotic. They were able to get good stuff from each other. And so evolution allowed that bacteria to become part of that cell permanently. This is going to be, we'll talk about this in more detail. The case with a lot of our organelles show evidence that this is true. It's pretty interesting stuff. So evolution, we think that. We also think that a lot of these microbes shaped our atmosphere. We didn't have oxygen. There's actually evidence that the atmosphere didn't have oxygen for a very long time. We think microbes are the ones that made enough oxygen to change our planet and allow for larger life to exist. Pretty cool stuff. So we have, that would be through photosynthesis. We know using light to make energy, and then their byproduct is oxygen. That's the process they would have gone through. We know that plants do that, but most of the oxygen on our planet is in fact going to be coming from microbes. This includes algae and, you know, plankton and stuff like that. So don't forget those guys fall into this category. Yeah, we already said this. Microbes, of course, are going to be important for decomposing things. That's why it smells bad when things are decomposing. Rotting, everything like that is driven by the microbes that are breaking down the tissues and the fluids and the makeup of whatever these things are that are dying. It doesn't have to be animals. It can be plants. It could be whatever organic material. So they're important for that. We know they're important for our soil. They live in our water. They can be involved in our atmosphere as well. We learned about making the oxygen, but they can be involved in making methane as well. Methane is actually a worse greenhouse gas than carbon dioxide even. And bacteria are usually involved in making methane or archaea. And anyways, so gas production by them, microbes living in the Earth's crust. So the soil Microbes are essential for helping plants be able to use nitrogen. And as we learn about the chemistry of life, we should know that nitrogen is one of the most important chemicals in life. So your DNA, that's gonna be nucleic acid. Those ATCG that make that DNA that is the code or whatever, those are called nitrogenous bases. So we need nitrogen just to have DNA. And if we talk about proteins, what are proteins made up of? Amino acids, okay? Amino means NH2, that's an NH2 group. So that's a nitrogen. So already we've got two extremely important biological molecules that have to have nitrogen, and we can't get our nitrogen without eating some other organism. And those organisms that we eat, like plants, they can't get their nitrogen without bacteria turning it into a form that they can use. And without those bacteria, the plants can't grow. We've had a huge problem with that lately because of global warming, getting too hot for the bacteria to be able to live in our soil. And so the plants can't grow because there's no bacteria there to provide them with nitrogen. This is a huge problem in places in the tropics right now. Like Sri Lanka, they're going through a huge crisis right now with food because they can't grow anything because their plants just can't grow anymore. So it's terrifying to think about. That could be a thing. Anyways, we know we can use microbes for a lot of things. Making our food, for one thing, like beer, cheese, bread. We know we can use it for getting our antibiotics, like mold. We know penicillin comes from mold. And then we know biotechnology stuff. So did you know that insulin nowadays, the reason it's so affordable and effective for people with diabetes, that's why they can just live in society nowadays. I don't know if you guys knew this, but there's like a deadly sort of diagnosis back in the day before we had insulin available. When we first had insulin available, it came from cows. So not nice, that saved people's lives, that's great. But now we can actually make insulin by putting the gene for insulin into bacteria, and the bacteria just crank it out for us. Isn't that cool? Like, I think that's pretty neat. So this is saving people's lives at an affordable rate, basically. So it's neat what biotech can do for us. And yes, we will talk about GMOs and stuff later on in the course. If you're interested in that stuff, we'll have that big argument. Okay. All right, pathogens. A pathogen is any agent that can cause disease. Disease is a deviation from health. That's all disease means. If you're deviating from what is typically considered health, then that is a disease. And if an organism is causing that disease, that organism is called a pathogen. Okay, so that's what that is about. Some diseases are going to be, carried by pathogens. They're communicable. They are infectious. Whether we're talking about infectious diseases like dental caries, so tooth decay is the most common infectious disease in humans. That shouldn't surprise anybody, right? Disgusting, but it's the truth. It's the most common infectious disease in humans. It's not necessarily communicable to a degree, right? As long as you're brushing your teeth, you're pretty much gonna keep it at bay. But then we have the communicable ones like a virus like COVID. That's communicable, obviously. You can transfer it from person to person. But you have other diseases that are not any sort of, germ at all. No microbes involved. Things like cancer, things like diabetes. These are diseases. So don't forget that there are diseases that are not caused by microbes. They can just be diseases on their own. Any deviation in health counts as a disease. We're mostly, of course, in this class focusing on the ones caused by microbes. Don't forget. Obviously, heart disease here, no surprise that heart disease is going to be the #1 disease in humans. Yellow, that COVID-19, I think that's the only one on here. Oh, we got some lower respiratory diseases and influenza down at the very, very bottom. But COVID-19, look at how high that is. That's an infectious disease, but cancer and heart disease are still beating it out. Those are pretty serious, going to be killer diseases. All right, so let's talk about some of the characteristics of these guys. Archaea, eukarya, and bacteria. We kind of talked a little bit about how bacteria and archaea are so simple. They probably existed earlier on in evolution, and eukaryotes evolved after that, dependent on them. We have some organisms that are single-celled. All bacteria and archaea are single-celled, all of them. Sometimes they exist in biofilms. I'm sure you guys heard about that. You guys know about the plaque on your teeth? I know, going back to that. You guys are gonna want to brush your teeth after this. The plaque on your teeth is a biofilm. It's just bacteria that have kind of like, grown together and created this film on your teeth. And you have to scrape it off periodically to get it off, or brush regularly to keep it from forming in the first place. And that's why flossing is so important, because you can't brush in between your teeth. You've got biofilms forming in between your teeth. And that's why we floss. I bet you guys want to floss now too. So anyways, that's a biofilm. Well, it's not a multicellular organism still, right? It's still single cellular organisms. They're just working together in their environment. Okay, so always bacteria and bacteria and archaea, A and B, these guys are going to be the single cells. The eukaryotes, the ones that came later in evolution, they're the ones that can become multicellular. Okay, so think about them as being more complicated. Now they can also be multicellular. Not all of them are, but many of them are. Some of them are single cell. But yeah, bacteria and archaea always single cell. The bacteria and archaea are always smaller, like 10 to 20 times smaller. So if you were to be looking in a microscope and you knew that one cell that you saw in your image was a bacteria and one cell was a eukaryote, you could tell by looking at the size probably which one was which, just by the size. So there's that sort of indicator, as well as we don't have any membrane bound features inside of the bacteria. There's just enclosed and all the goo and everything floating around in there, just floating around, and it's not contained in any way. Whereas eukaryotes, you will see those organelles, nucleus, mitochondria, chloroplasts, endoplasmic reticulum, all those guys. And then the acellular microbes, viruses, and prions. If you were to take a sample from a patient that's coming in with diarrhea, let's say, right? And you culture their diarrhea or look at it under a microscope, let's say. You do a stain and you do not see any cells. Now, of course, they'd have their own microbes that would be living in there. But let's say somehow you're looking just at the pathogen, right? And there's no cells at all in there, then that person has diarrhea caused by a virus. You would know that immediately. Okay. If you saw cells, just because they're cells, can you tell the difference between it being a bacteria or a eukaryote? No, right? You would have to look even further. Does it have organelles? So if you see cells with organelles in there in the samples, what's causing it, then you know it's a eukaryote that's causing that disease, right? So that's one way you can kind of break that down. So here's some images just of these guys. I'm not going to go over them really right now, but we can see this up here at the top. Let's see, this is so hard to see on here. Here. Up here at the top, we have Tinea solium. This is a tapeworm. So that's the head of a tapeworm, pretty nasty looking. I'm not going to go over all of them, but some cool stuff that we can see here from the fungus, the protozoa, and all the way down to the prions, which FYI are just proteins. We'll get into them in chapter 7. So in order to look into science in general, we know the scientific method. I'm sure most of you guys have heard about this before. This is, generating a hypothesis, which is a guess based on what you've been observing. So you look at all these factors going on, and you're like, I bet that this is why this is happening. Okay, I'm going to go ahead and now devise an experiment to test whether that is true or not. And if it turns out that your hypothesis wasn't true based on that experiment, do you need to change the experiment or do you need to change the hypothesis? Well, you can decide on that as based on whatever your goals were, right? But that's a scientific method. A hypothesis is that educated guess based on what you've been observing, and then you can test it with experimentation. That's really all you need to know about that. Yeah, so experimentation supporting the hypothesis, but we're not, if it did, you went through your experiment and it supports your hypothesis, that doesn't mean that your hypothesis is fact now automatically. We need to probably do several other tests to approach it from different angles. We need to have other people maybe do the test to verify it. And even then we might only develop a theory. And that's why we don't see so many laws out there anymore. We see a lot of theories because it's just hard to say that definitively always, yes, this fact. That's the idea with that. All right, let's talk about Louis Pasteur. Louis Pasteur came up with the concept of germ theory. Okay, so this is really revolutionary for the time. Back in the 1800s, people believed still that disease was caused by miasma, which is bad smells, or can you imagine if you go into the bathroom after your husband and getting sick from that? Anyways, so either by bad smells or they thought it was just spontaneous generation. That was their concept. So that just meant hey, they noticed that they left out a piece of steak out here on the countertop for days and then maggots appeared. No, we know the maggots don't just appear out of nothing. Probably a fly came by and laid some eggs on it. That's where the maggots probably came from. But if they don't see that, or even if they see the fly on there, they didn't see the eggs probably. They're hard to see. So why would they attribute it to the fly? So they thought things would just come out when things were going bad, things would just spontaneously generate to get rid of, start rotting it away. Or whatever. So that's what they thought. Pasteur thought that that was a load of crap. So what he did was he took these flasks. And this one was a swan flask, and one he broke off the top so that it was just a regular straight up and down flask. And he had boiled the media in each of these to make sure they were free of microbes. And then he just set them out and then saw what happened. Well, turns out microbes can get in more easily with that one that's broken off than it can with the swan flask. It's hard for them to actually migrate through the atmosphere in that little swan neck there. So it turns out if they didn't both grow anything, right? If it was spontaneous generation, they would have both grown something. Wouldn't have mattered if there was a swan neck on it or not. Both got air coming to them too. But it's just the microbes can't move easily through that swan neck. So that is Pasteur's experiment that first showed us germ theory, the idea that there has to be something there in order for there to be growth, right? So if your media turns cloudy, which is what happens a lot of times whenever we're growing bacteria, if your liquid media turns really cloudy, then something is growing in it. And often gets a smell too. And you guys will be real familiar with that in lab. But yeah, so it's the sign that something's growing in there. But this other guy, it stayed clean, showing that there had to be something introduced in the air, via the air at least, to contaminate it. So here's some other guys that kind of supported his thoughts. Robert Koch, he had a specific, he had steps that he would go through to link specific microbes with specific diseases. We'll talk about his experiment later on in the course, but that's what he came up with. He actually identified anthrax this way. Next we have Dr. Holmes, I guess, and he noticed that women who gave birth at home had fewer infections than women who gave birth in the hospital. He at least acknowledged that, saw that as an issue, but Dr. Semmelweis is the one that sort of confronted it head on. He showed that women became infected in the maternity wards simply because their physicians were going in during autopsies and handling other patients and were not washing their hands before coming back and handling these women. They didn't wear gloves because they didn't think that was a thing. How could you, why would that get transferred? That didn't even occur to them that was a thing. So that idea that Pasteur came up with, that there's germs, and now that you are actually transferring these germs from person to person, has changed medicine completely. Lister actually took it upon himself to utilize the concept of hand washing and sterilizing operating rooms with antiseptics in order to prevent people from getting sick and dying from surgery. So in his surgical suites, he would treat them with phenol, is what it was, carbolic acid. Sure. So they're taking pictures, just FYI. So they would treat it with carbolic acid, and that's just phenol, and that would kill pretty much everything in that area. And they would be sure they were like basically scrubbing in every time, and that has significantly reduced the amount of infections that they saw associated with, contact basically with that. So that is revolutionary. Lister, yes, this is Listerine. So originally Listerine was a solution that had phenol in it. It doesn't anymore. So phenol is pretty dangerous for people. But so yeah, Pasteur, he also invented pasteurization, which involves stepwise heating of liquids to try to kill microbes without destroying the integrity of the liquid itself, like milk. We pasteurize our milk, right? So that, anyways. All right, if we're going to name and identify and categorize your microbes, because why wouldn't you? Because that doesn't make things complicated or confusing at all. We have special kinds of nomenclature that we're going to use to talk about our microbes. And it's different from bacteria to viruses and eukaryotes, because why wouldn't it be? Again, so we want to be able to classify them and identify them, and then we can say, okay, this is Escherichia coli or E. coli based on these traits that we've seen, and you have a name to ascribe to it now. Say that it is this. We've named them, of course, after a lot of people for the most part. A lot of them are named after people or after features of those organisms. So they're not unified in that. It would be nice if they were. All right, so this is the binomial system of nomenclature. Like I just said, Escherichia coli, right? Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae, whatever. Those all have two names to them, right? All of them do. Even if we're talking about Salmonella, you're actually talking about Salmonella enteritidis is what you're talking about. So they all have two names. That is binomial nomenclature. So here's how we write this. It's always going to be capital letter on the first word, lowercase letter on the second word, italicized when you're typing it. If you're writing it by hand, it's supposed to be underlined. Okay, so when you guys are turning in your unknown report, which I know that's like not even probably on your mind right now, in the lab, all of what we're doing is trying to identify what bacteria I'm giving you. So I'm going to give you 2 different bacteria, and each table is different actually from one another. So it's your, responsibility to figure out what your 2 bacteria are for your table. And when you enter in your name of your bacteria, when you do have it correct in your form, I expect you to use binomial nomenclature, for example. So it's probably one of the only ones that you're ever going to encounter where I'm going to check that. Because all of our exams are multiple choice. So I don't want to see your handwriting. I don't want to see you try to spell things out. To me, it's not as serious of an issue that you know how to spell things correctly, because a lot of the scientists don't even spell it correctly anyway. But that you recognize things, but I do have to test you on it. So that's going to be the one time, and I will remind you about it when we get closer. So no worries. But if I ask you about it on the test, I'll give you choices. It'll be like, some will be 2 capitals, some will be italicized, some won't. So you'll have to know based on looking at it which is correct. Be aware of that, how to correctly write these things. If you can remember S. aureus, where the S is capitalized and the A is little, or E. coli, if you can remember the E is big and the C in coli is little, then you got it, man. That's all you really need to know. Just remember it's italicized. Okay. Taxonomy is boring, and it's one of those things I feel like it's like almost useless, but I have to teach this to you guys. It was developed by Carl von Linné. It's just a way for us to categorize organisms into groups that we're studying, so we can look at similar features between them, right? You guys remember this from grade school? Okay, so I do need you guys to know it in order. Get whatever, mnemonic or whatever you like in order to remember it, like King Philip. I don't even know what the thing is that people use to memorize it. I just know it by now because I've said it so many times, but kingdom, phylum, class, order, family, genus, species. Whatever you need to do to get KPCOFGS. That's the important one. Domain, I have that one at the top. Yeah, it's bigger than all of them. Domain only includes eukarya, bacteria, archaea. That's it. So that's all domain is. But obviously the other ones are going to get more complicated. And we think of them more in the context of eukaryotes typically. So if we're talking about Animalia, right? Animalia is a kingdom. So, all right, it's usually gonna, when we look at phylogeny, we've applied a lot of phylogeny into categorizing these guys. Phylogeny is just using genetics to branch them off by relatedness. Okay. Oh, we were talking about the domains. We can use small subunit of the ribosome. But anyway, everything has ribosomes if it's a cell. Okay. Viruses, they're on their own, man. But if it has a cell, so if it's bacteria, archaea, or eukaryotes, then it has ribosomes. These are our machines that make our proteins. So we can compare how similar the ribosomes are to each other because everything's encoded by your genes. Literally everything in your body is encoded by your genes, including your ribosomes. They didn't come out of nothing. So the difference genetically between them, we can see how related things are using that because everybody has them. Kind of neat way to do that. All right, so here we go, guys. When we do the question, we're all going to stand up for this because this is it. Like I know everyone's like, oh, I'm not standing up. Yeah, you're going to be lazy and sit down. Exactly, that's my point. That's why we're standing up. So it's going to wake up our minds, get up, and also make it so you can't look at your notes. If you're paying attention. If you're paying attention. Yeah. So like I said, you might get the answer wrong, but even wrong answers teach us something. Because if you tell me the wrong answer, that gives me the opportunity to tell you guys why it's wrong. And if you get it wrong, guys, I'm not kidding. Number one, nobody cares that you got it wrong. Nobody's judging, right? Would you judge somebody else if they got the question wrong? Nobody cares. So don't worry about that. Nobody cares if you got it wrong. Number two, when you get it wrong, you're probably not the only one that would have guessed it wrong. So probably more than one other person would have guessed your wrong answer. So please, this is not meant to shame anybody. This is meant for us to learn from one another and see where our weaknesses are. That's what this is. Okay. All right. They're not meant to be too difficult. So what exactly, I'm going to call a name. What exactly is a pathogen? I have Leanne C. All right. What's a pathogen? Make a guess. You're good. What does it sound like it is?
Something that will infect other cells.
Good. Okay. So I will accept this answer. That's fine, right? So it's any microbes that cause disease. Okay, good job. So that is delta. Delta, you guys just got to know. All right. That's all this is, guys. So it's okay. Don't stress out too much. It'll get easier as the semester goes. So next one. What is, in a nutshell, what is germ theory? Sarah F. Just basic. Just basic.
Mhm.
That's what we're talking about, where we're saying germ theory is just the idea that disease, anytime we see disease or rotting or anything like that, that germs cause it. That's all it is. So nothing, so don't overthink it. Yeah,
Okay.
yeah.
I think it's
Alright.
flying like Minecraft.
All right, next one. All right, so try to keep your answers to yourself because you guys might be in different groups. So I can't assign a point to a group now. You're in different groups. Do you know what group you're in? You don't know what group.
I forgot.
Well, Sarah, you're in alpha. So. Yeah. All right, next one. Like I said, there's only 5. So bear with me. All right, can you name one non-cellular pathogen? So that means a pathogen that isn't made of cells. All right, Ariana A. The helminths?
Yeah.
Okay, so I like where you're thinking with this, right? I get where you're coming from. So with the helminths, they can be bigger, right? They're not necessarily microscopic. So I get where you're going with that and where your mind is with it. For non-cellular, I'm looking for anything that doesn't have any cells at all. Helminths are eukaryotes. So they do have, anything big like that is going to have cells, right? So she is part of Bravo group. Anybody else in Bravo? Yes. Anything that doesn't have a cell. You can name a specific one. Yes, but yes, virus is good. so yeah, yes, flu, that's a good one. Yeah. But yeah, you were right with virus. That was the correct answer. I don't know why I made you say a specific one. Yeah, so yeah, viruses don't have cells. That's what we're looking for with that one. Okay, real basic. You guys probably overthinking a lot of this stuff. But you will probably do the same thing on the test. That's why we want to go through this and make it easier for you guys to think about this stuff before the test, right? So, all right, list the levels of taxonomic classification. Let's just tell me, if you guys can, whoever this is that I picked, tell me the first one, not domain. What's the one after domain? Renee D. Okay, fair enough. At least, yeah, you tried. Do you know any of the other ones? Any of them? No. Okay, yes. So it was kingdom. That's exactly what I was looking for. Good job. Is that it? Yep. I think that was it. Was that 4 questions? Yes. I just didn't write this one, but that's fine. Whatever. So let's move on.
It's okay.
Yeah, I think we'll be okay. You guys can sit down now. Thank you for being good sports. But that's what we're going to do when we do the questions. I'm hoping that it helps you guys kind of get your minds working a little bit better to stand up. And that helps like break our zoning out. So that's the point of this. So in chapter 3, the first thing we're going to talk about is the 5 eyes. This is one of the biggest concepts as far as investigating what microbes are causing what, especially with the bacteria. It's pretty much our bread and butter when we're going to be doing stuff in the lab. There's 5 of them. The first one is inoculation. That means introducing the microbe to a media. So if you have sterile media, it's boiled, it's autoclaved, whatever it is, and you're putting a sample onto the media, whether it's from the back of your throat, whether it is from, a sample that I'm giving you guys, whatever it is. But if you are now turning that sterile media and putting a sample on it, that's inoculation. Okay. Inoculation. Incubation, that's just putting it in the proper growth conditions that it needs to grow. If I were to put, swab your throat and then put it on a media and I put it in a refrigerator that's cold, is that going to grow? No, so that's not the proper growth conditions. That's exactly what this is talking about. Probably around body temperature. It's usually ideal for most of the pathogens, okay? Isolation, just separating them. If I swab your throat, do you think only one bacteria is going to come out of that? No, it's going to be like a lot, right? We all know there's bacteria all over us and in us and whatever. If I take a stool sample from somebody, how many different kinds of bacteria are going to come out of that? A lot, right? So how do you separate out the one that you're interested in? That's isolation. Inspection, that is like now we've isolated it. We've got it working with a pure culture of just like that one bacteria from that poop sample. Now we're going to be looking at it in microscopes to see what it looks like, doing different stains to see how it stains, and then also growing on different media to see what kind of media it can grow on and different things like that. Okay, that's like all lab is going to be. And then identification is using all of that information and putting it to compiling it together and saying, oh, well, if it grew on this media and it didn't grow on this media and it changed this media this color and it stained this positive and that one negative, that means it's E. coli. Okay, that's what we're doing in lab, y'all. Okay. All righty. Producing a culture. A culture is just talking about any propagation of microbes in a media. Media is whatever the microbes are growing in or on. So don't think too hard about that. Medium is supposed to be the singular. I almost never call it medium. I will almost always just call it media because I don't know. That's how, I know it's grammatically incorrect. I don't really care though. So it's just how this is going to be. Inoculation, like I said, putting microbes onto your media to culture it. And then sterile is talking about anything that's free of all life forms. In chapter 11, we'll talk about the difference between sterile versus like decontaminated or disinfected. Those are not the same thing. But sterile means free of all life forms. Okay, so let's talk about media that we can grow on. Your media can be defined by its physical state, its chemical composition, and what it does for us, right, or for the microbe. So physical state, nothing crazy here. Liquid, semi-solid, solid, or solid that cannot be liquefied, whatever. I just think it would be more happy if you guys even just knew it was going to be liquid and solid. And sometimes the solid is less solid, like a semi-solid. Okay. Really what we're going to be working in lab is liquid or solid for the most part. We will have some semi-solid, but that won't be way later. Chemical composition, chemically defined or it's complex. If it's chemically defined, that means synthetic. That means you know exactly the amount and concentration of every chemical in it. Some of the media that we use, like blood agar, we don't know the exact concentration of every little chemical in sheep's blood that we use to make the blood agar, right? So that is a complex media. We just know it has sheep's blood in it, but do we know the exact composition of the chemicals in that blood? No, and that's fine. So that would be a complex medium. And then functional type. Are you just growing crap? General purpose, right? You just need to propagate it somehow. Enriched means we have an extra thing added to it to help it grow. When we talk about what that means in a minute. Selective means we're selecting for one thing and maybe not another. So selects for some things to grow and not others. Differential, telling the difference. Reducing has to do with anaerobic, things that can't live in oxygen. Specimen transport speaks kind of for itself. If you've ever been at a hospital and they swab and they put it back in that little tube and then they're going to mail that off to DLO or whatever, that has some media in it that you'll usually pop it. It's a little thing that breaks inside and it's liquid to help the stuff, whatever you swabbed, be happy until we get it to DLO. Okay. An assay for the actual media to be done in that specific, media. And then enumeration means counting the cells. So there's all sorts of different kinds of media that we can use for all sorts of different purposes. That's the point. Okay. All right, we already know what liquid and what liquid and solid means, right? Liquid, it's liquid, it's liquid, it's like broths and stuff like that. Semi-solid, it's a solid, it's like Jell-O. Okay, that's what semi-solid is. Solid, it's gonna be literally like, it's not super hard, it's not hard like this. It still has a bounciness to it. It's like a harder Jell-O. And that's like agar, what we typically will use in lab is solid surface agar. They're a little soft, but they're not semi-solid. They're not almost liquid, right? Agar literally is a term referring to the algae that we use that helps solidify this. So it's like an extract from the algae that makes it semi-solid, basically. And how much you add into it makes it more solid. Okay. So remember that. Remember, agar is from algae. All right, we already talked about this synthetic versus complex. We don't need to go back over this. Yeah. General purpose versus enriched. So let's look at that. We already talked about general purpose. Just grow whatever on it. That's all that is. Enriched media. This is where we grow fastidious organisms. A fastidious organism is any organism that needs something added to its nutrients in order to survive because it can't make it for itself. That should sound familiar to you guys because that's us. If I take away all the vitamin C in your diet, what happens? You will get scurvy and eventually you will die, right? If I take away your B12, you're gonna have some serious health issues. Your body can't make B12, your body can't make vitamin C, and there's other vitamins that your body can't make. It relies on your diet to get those supplied to your body, right? So those, a lot of times, so it's vitamins, that's why they're called that. So we are fastidious organisms because we require these things to be given to us because we can't make them for ourselves. Those things, like our vitamins, those are growth factors. Now, bacteria don't necessarily need vitamins, but there could be other things that they cannot make for themselves that we have to supply for them. Those are still growth factors for the bacteria, right? So we are fastidious, we need growth factors. Some bacteria are fastidious, some of them need growth factors, okay? And then when we add that into the media, that's called enriched. Okay, so this is a pretty good example of selective media versus differential media. So if you have a mixed sample, like you swabbed somebody's throat and you grew it in some broth, and now you know there's a whole bunch of bacteria in there, right? But you want to look for strep because you're pretty sure strep is the problem here. So you'll plate it on, if you use general purpose media where anything will grow and all of the different bacteria species will grow. But let's say you use a selective media that selects for strep bacteria. Now, anything else that's contaminated in your sample will not grow, but anything that is strep will. So that's selective. We're selecting for a group of bacteria, right? This other one on the right is differential. I'm sure the next slide is going to say it. But here we have our mixed sample, and we'll plate it on general purpose media, same exact result. All these different bacteria that's in the sample are going to grow. In differential media, though, all those same bacteria are still going to grow, but they might change colors based on how they react with the media. They might make the media itself change colors and whatever. So now you can tell the difference between them, okay? So that's going to give you information about your bacteria too. So you can select for them, or you can plate them on media that lets you tell the difference between them. Okay, that's what selective and differential media are. So differential, we have some color change. This stuff up here is one of the media that we're going to be using in lab later on in the semester, TSIA, triple sugar iron agar. And this is all different things that you can see happen. So this would be uninoculated, original. And this one has black in the bottom, and this one has got lifted up and it has bubbles in it and it's yellow. Like, that's crazy, right? So those bacteria and those second two tubes are clearly different from one another. And that's information you can get if you know what the media is and what you're looking for and what those results mean about what they can ferment and whether they can reduce sulfur or whatever else you want to know about the bacteria. And that information you can use to help figure out what is what. Okay. Differences in colony size, colony colors, media color changes, all that stuff. It's usually color change. It's usually color change that's going to be involved in differential media. The idea is that most organisms should be able to grow on the media, but you're just going to see a difference in color, basically, based on their metabolic byproducts normally. You can also have media that can be both differential and selective. But first, blood agar. Blood agar, like I already mentioned, it's just agar. It's like a regular normal agar with blood in it, sheep's blood. And what we're looking for in blood agar are hemolysins. And these are enzymes that are going to break down the blood cells in the blood agar. If you are able to break them down completely, then you will see a clear area around where the bacteria are. So that is a beta hemolysis, or hemolysis, I guess you could say that. Complete lysis of the red blood cells. Alpha hemolysis, it's like it's going to turn it green. You ever seen that sort of like whenever you have blood smeared on something and it'll get sort of green tinge to it? It's sort of like that. It'll have a greenish tinge to it. And that's alpha hemolysis. So it's incomplete. We're breaking down the red blood cells, but we're not breaking down completely. That's why we get clearness around the beta hemolysis. We get green around the alpha hemolysis. And for some reason, there's something called gamma hemolysis, which means that you're growing, but you're not showing alpha or beta. It's like you're just growing. Somehow that's hemolysis. I don't know why it has a hemolysis name, but whatever. So this is growth without any visible hemolysis. Okay. All right, so here's some pictures of that. This is gamma. We don't have any real change. We see growth, but it didn't change it. Alpha has that greenish tinge to it, but you can see quite clearly for strep how it has caused clearing around where the bacteria are growing. You can see that very clearly on even in the picture. So now you can say comparing Enterococcus and strep sanguinis or whatever, and then strep pyogenes, that our patient A, if we're just comparing those three, probably has strep pyogenes. Okay, that's the strep that causes strep throat. Okay, like I said, some media can be both selective and differential. We can talk about McConkey agar. We can talk about mannitol salt agar. Mannitol salt's probably easier. In mannitol salt agar, like it says in the name, it has salt in it. So that high salt concentration favors the growth of Staphylococcus bacteria. So gram-positives, which we'll learn about why that is the case later on, but gram-positives like staph, they have the ability to resist salt, like on your skin. So when I say like your skin is salty and they can resist salt, and I'm talking about staph, that shouldn't surprise anybody, right? We know staph grows on skin. So staph can grow on the salty environment of the mannitol salt agar. So that's selective because not everything can grow on that, right? So gram negatives, like E. coli and stuff like that, can't grow on the saltiness, but things like staph can. So it's selective. We also have color change going on. Maybe there's all these different kinds of staph that can grow on the mannitol salt agar. What if we want to tell the difference between them? Staph aureus can break down the sugar mannitol and create an acid byproduct. When that happens, the acid creates a pH change in the media, and that causes the color change in the media that we see. So the yellow and the fact that it grew, those two things indicate to me that that's Staph aureus. Okay, if we had Staphylococcus epidermidis, the most common staph on your skin, then it would not turn yellow, but it would still grow. Okay, so now we've got things we can tell the difference between organisms based on how, if we're doing selective and if we're doing differential, and then if we're doing both at the same time, we did here. All right, so isolating our bacteria, I'm going to look at colonies. Colonies are discrete mounds of cells. You know, you guys have seen pictures. I only just showed it friggin' on that other slide. Plates, when you get the little dots on them, that's a colony. Okay? One dot is a colony. It is formed from one single cell multiplying up. So one colony came from one cell. All right, this is the streak plate method. So if we want to get to one colony, because if I'm telling you that one colony comes from one cell, that would mean that if you take a sample of that one colony that's all by itself, you could create a pure culture with that, right? If we can get them separated out like that. So how do we get them separated out like that? One of the best ways we can do that is the streak plate method. It's one of the first things we're going to be doing in lab. Okay. You take a plate and you're basically going to be spreading it out a little bit, turning it, spreading it out, turning it, spreading it out, and then turning it and spreading it out some more. Each time you're turning it, you're sterilizing your instrument, your loop. And so each time you're turning it, you're diluting out your sample more and more and more. You're spreading it out further and further and further. The idea is that at the end, you'll have these individual colonies that you can pick this individual colony. If you take a sample from up here, are you picking up just one kind of bacteria? Probably not. But having them individual like that at the end, now we can create pure cultures with those colonies. Make sense? One colony by itself is going to be basically one cell growing up. So you should be able to create a pure culture from it. All right, so what is a pure culture? It's just a culture that has only one known organism. And that's really the only term that I really need you to know. I'm not going to ask you about anexic, but what anexic means is just a term referring to being free of other living things in general, other than the thing you're studying. So that would be, what we would describe as pure cultures as being. Okay? A subculture would be a second level culture from a well-isolated colony. So if you had done your streak plate and you've got all these little dots out, right? And you take out one of those dots and put it into a broth to grow up a pure culture, that's a subculture. That was your first culture. This is your second culture, that's your subculture. All right, mixed cultures just means you have more than one organism in it. And a contaminated culture means that you had it separated out, and now some other crap got in it. So those are different things. So if I take a swab of my throat and I put it on a plate and there's a whole bunch of stuff growing, that's a mixed culture. But if I had separated that out and got only the strep bacteria, and then later I saw some, they might have white colonies. I saw some yellow colonies growing in there randomly later. Uh-oh, right? So that's contamination. So remember, those are different things. All right, different ways that we can determine the differences from one bacteria from another. Phenotypic, so that is going to be things like looking at the media, what can grow on what color change do we see. Genotypic is literally looking at the genes of the bacteria. Okay, so DNA testing, PCR, stuff like that. Immunologic, now we're talking about antibodies, which yes, in this course we will be learning about the immune system and antibodies and what they are and what they do, but they can be used as a tool for now, that's all you need to know. Macroscopic analysis versus microscopic analysis. So looking at the culture and the colony colors and all of that versus actually looking at the cells in the microscope, doing the slides. And, okay. Biochemical tests is basically just what I'm talking about with these other ones. If it can break down a material or not, right? So telling you about how its metabolism works. Can this organism use citrate? Can this organism use malonate? Can this one ferment glucose, or this one ferment lactose, or whatever else? So you can ask these questions about your bacteria, and that's dealing with their biochemical makeup, their metabolism, stuff like that. So that's what this is talking about. It can tell you if something were to change a color in a certain kind of media, could give you the answer of what kind of enzymes must be there if it did change color. If it did grow here, then it must have these things going on in the cell. So these are not just going to tell you, okay, yellow, that means E. coli. It'll tell you that yellow means, oh, this enzyme is produced so we can break down this thing. E. coli has that enzyme. Okay, so that's more information there than just saying color change. Okay, we know about genotypes. That's just going to be looking at DNA. All right, well, we already talked about how eukaryote cells are in general bigger than bacteria by about 10 times. I'm not going to go through this whole thing, but if we were to look down here at HIV, this is HIV, and this is 100 nanometers versus 100 micrometers, 1,000 times bigger for Giardia lamblia. This is something that causes diarrhea. If you go camping and you don't boil your water, even if it looks clear, even if you got it from a spring that's all bubbling and it's, you know, nice and clean looking, you can still get Giardia. And this is, we're talking like 2 months worth of what they call greasy malodorous stools. So I don't know what to tell you about that. It sounds awful, but it is protozoa. It is a eukaryote, and apparently is 100 micrometers. 1,000 times smaller than that, we have HIV. Now you can't even see Giardia as it is. So that should put into perspective how tiny an HIV virus really is and compared to a eukaryotic cell. Pretty nuts. Look at poliovirus is even 10 times smaller than that. Polio is one of the smallest viruses that we know, that and parvo. And then this is a prion, which we're going to talk about in chapter 7, but they are misfolded proteins, basically. And that's all they are. They're just a protein that's misfolded. So of course it's smaller than viruses. So of course it's smaller than anything else because it's just a protein, and viruses are made of proteins, you know, so linked together. So yeah, prions are much, much smaller. If you guys don't know what a prion is, like I said, we're going to go over in chapter 7, but an example that most people are familiar with, mad cow disease. Okay, so mad cow disease is caused by a prion, a misfolded protein. Okay. All right, magnification. We can, like, I don't know, bend, like lenses that are curved can help bend light to help make images look larger. That's magnification. Refraction is talking about the way that light will bend when it goes from one material to another. So we can use that knowledge of, okay, if glass is so much different than air, and as we're passing from our glass slide into the air and then into another glass lens, there's going to be refraction. We're going to lose some of those light rays that told us stories about our sample. So if we put something in between there that is more similar to the glass than the air is, that can reduce that refraction. So we can keep those light rays that told the story about our specimen in where they're supposed to be. Okay, that substance is called immersion oil. Okay. All right, anyway, so you have your slide on your microscope stage, and the light is going to shine up through the slide, right, from the condenser and through it and then into another lens. That's going to go through, so that's going to magnify it. That's going to go through some mirrors and then out through what you're actually looking through, the eyepiece. It's another magnifier there. Okay, so that's how all of that works to make it so where you can see it. So if you're looking at the thing that's rotated on the 4x, well, if you were to look straight at that, that would be 4 times bigger than reality. But if you look through the lens, that's 10x. Now that's multiplied. So that is 40x overall, so 40 times bigger than reality is what you would see with the 4x and then the 10x thing, okay, to multiply it. So our microscopes are the same way. So if you have it set on your 10x on your actual objective lens, and then you're looking at the eyepiece, another 10x, that's gonna be 100 times, okay, the actual real thing. That's what you're looking at. We're not gonna go too much over the parts of the microscope right now because we're gonna go over that in the lab, okay? It makes more sense there. When we talk about this contrast, I'm sure you guys know what contrast means. It's just being able to see something more clearly. If something is gray on a gray background, you can't see it very well versus like black on a white background. You can see a lot. It's more contrast, right? Okay, so our objective lens. So we had our specimen here, here, and the light coming through it, and it's going to hit this first lens here on our objective lens. That's our first magnifier right above it. That is our real image because it's the first thing to see it, the first thing that's coming in contact with real image. Then that's going to get mirrors and another lens. We're distorting the image even further. So then when you look at it, that's the virtual image. Okay, it's been messed up more. Just think about it that way. So it's virtual, it's not real anymore. So that would be how that would work. And here's just how the light travels through everything and creates whatever images. We already talked about magnifying power. 100x is where we will be using that 100x objective lens. It uses the immersion oil to reduce the refraction, so we can see better resolution. Resolution is talking about clarity of the image. So like I said, there's the oil on the right, and it's showing us that the rays of light are being maintained and actually sent properly to the lens. So that's what the oil does for us. We talk about that contrast. So preparing our specimens depends on what you're going to be looking at with your specimen. Could be you want it to be dried, could be you want it to be wet. Literally depending on what you are looking at on your specimen would be how you would determine what you need. If you're looking at your specimen as being modal, if you want to know if it's modal or able to swim or not, then that motility is going to be something you want to look at in a wet mount with living specimen. So that's why you would want a wet mount, but it can dry out. It doesn't always stay moist the whole time. With fixed stains that are dried, this would be a smear technique. You're literally just watering down your sample and spreading it out and literally just letting it to dry. A lot of times we're also going to be heat fixing it. So passing it through fire it kills everything, but it also adheres things to the slide a lot better, which is like our main purpose of what we're doing. And it maintains the structure of the cells and what is in them, so preserves them. Then we can stain those smears, and then we can use basic or acidic stains. Basic stains, I would really rather, because I'm not going to really so much ask you about basic or acidic. I will ask you about positive versus negative stains. Okay, positive stains. Our cells have a negative charge on the outside. That's from the phospholipids on the outside of our cells. The phosphate groups are negatively charged. So typically in chemistry, these opposites are going to attract each other. Positive is going to attract negative. So if our cells are negative, the positive stains are going to be attracted to that. So positive stains are going to stain the cells themselves. Negative stains, because they're negatively charged, are going to repel the negative on the outside of our cells. And that means that they're going to stain the background. Okay. All right. Yes, we just said this. So this is some pictures. You can see like the cells here are actually stained. You can see the actual cells are purple. Yeah. So that's clearly an example of the cell being stained by the positive stain. Here we have a purple background. That purple background is made by that negative stain. It doesn't make purple just on its own. You had to add something to it to make a background. Okay. So that's our negative stain. So therefore, we will be using both of these in the lab. Simple stains are easy. It's just you're staining your cells with one dye just to see them. They're usually not telling you really anything different about them, but just so you can see the cells. Maybe their shape, something like that. Differential stains, however, now we're using usually two different stains in order to tell the difference between the cells. Okay, so it's just like with our differential media, where we could see the difference from the color changes. Similar kind of thing going on with our differential stains, but at a microscopic level. So that would be our gram stain, acid-fast stain, endospore stain, capsule stain. We're going to look at all these in the lab very early on in the first few weeks. We're going to do all of these. And we're going to talk about more of these in detail in chapter 4. So I'm not going to go into the details too much right now. But it does want me to tell you that our gram stain, when we do this differential stain for the gram stain, is probably our most important stain in bacteriology. We're looking at bacteria and trying to tell the difference and figure out what they are. Your very first thing that you're going to be doing is a gram stain. Is it gram positive or is it gram negative? If it's gram positive, that means that the staining procedure, the gram stain, was positive. It went through retaining the stain and whatever. So it was positive is going to be purple, P purple, positive purple. Okay. Gram negatives. Negative to me, and it says pink, and I usually try to steer away from saying pink, red. Okay. It looks pink in the microscope, I understand, but if you can think of negative as being red is a negative color to me. Okay. So I think red, and I think that's negative, and then I think, okay, the gram negative stains red. And yeah, if you can tell the difference between pink and purple, you should be fine. And it's usually very obvious like this image here. Okay. You can tell which one would be red, whatever. Acid-fast stains are used to look at mycobacterium, like the bacteria that causes tuberculosis. They have a special molecule on the outside of their cells that you can identify with their stain. I'm not going to go into the details of it, but there are some other special staining techniques that look for endospores and capsules, other features that bacteria can have that make them more likely to cause disease and more hardy in the environment, and you know, can live for decades in the freaking soil and stuff like that. We can look for those characteristics with those. We'll be doing those in lab too. Endospore stain, looking for endospores. Like I said, we're going to talk about this in chapter 4. I don't want to get into the details of it right now. But these are just examples of differential stains. This is a capsule stain. You see how we've stained the background and the cells, and you see that white around them? That white is the capsule. It doesn't stain. So it's pretty cool looking stain. We'll be doing that one. And then we can stain for flagella, which are the structures that the bacteria use to move around. We'll learn that about that in chapter 4 too. Okay, we're gonna do this. No, maybe we're not. What time is it? Yeah, we got time. Okay, I'm gonna force you guys to do it again. Last time, all right? And then you guys can go. For real? All right, let's see. Cynthia D, what are the 5 I's? You can name one of them.
Isolation.
Good. Awesome. Isolation will take it. So we have like inoculation and incubation and isolation, and we have investigation and identification. All right, next one, selective media. This is going to be Ingrid H.
Oh. What is selective media?
Roughly, yes.
It is. Hold on, I know.
All right, anybody else from Delta Group know the answer? Yes. Yeah, so selective media, good explanation, and more specifically will be that media that will allow for some things to grow and other things won't, right? So we're selecting for some things to grow and selecting for other things to not grow. That's fine, you're good. All right, next one. Differential, uh-oh. All righty, let's see. Bridget H, I think she dropped it. Gavin A. No. All right, Madison H.
That just means that there's multiple organisms.
Yeah, so multiple organisms can grow on it, and also it could have different color changes based on whether they can use the media or not, right? So whatever's in it. So good. We'll do with that. All right, which part of the microscope generates the real image? Let's see. This will be liquid C. Do you know?
I'm not sure.
Yes. The eyepieces. Okay, so the eyepieces will be the virtual image. Does anybody remember what so she is Echo group. Does anybody from Echo know what the piece is called that's generating the real? It's not the eyepiece, it's the one by the specimen. Do anybody know what that's called? So close. Objective. Objective. All right. Good job. Yeah. I'll give it. All right. And then differential stain. Name one of the ones that shows the difference between things. that we just talked about and what it differentiates. So this is going to be Aubriana T. The ones that we just talked about here at the end, so I had like all those different ones that like, oh, we look to see if it is purple or pink, or we look to see if it has these weird things. Do you remember any what they're called? Okay,
I know what you.
I will leave you. Anybody from Charlie know what any of them were? Oh, sorry. Gram stain, that's one of them, right? So we had the gram stain, we had like the capsule stain, we had the flagella stain, we had the endospore stain and the acid fast stain. Any of those would have been fine, but the gram stain. And what it differentiates is it's going to be the basically the outer covering of those cells. We haven't really gotten into too much detail about that. That's okay. So I'm not looking at one of those. And I think that's it. All right, good job, guys. You put up with it. I'm proud of you. Be sure if you haven't signed in on the sign-in sheet that you do sign in on the sign-in sheet before you leave. It is back there on that back table. You have a copy to it.
Sorry. What?